Hey you guys, welcome to my YouTube channel. So in this video we will discuss about the Trypanosoma gambiense life cycle and pathogenicity in detail. So first we have habitat. Habitat means where its location, where it takes place. So it have initially found in the blood, then it migrate to the lymph, and final destiny is the cerebrospinal fluid of man. So here man is the primary host, we will see in the life cycle. So while its developing stages are found in the intestine and salivary glands of the some blood sucking insects, like we can see here the mosquitoes. Okay, so for example, mosquito here act as a secondary host. Primary host means sexually maturation takes place here. So in this secondary host, a sexual stages takes place. So here mammals like pigs, antelopes, buffaloes, reedbugs, etc. are the temporary or reservoir host. Okay. So first time it was reported by the Ballantyne in the blood of trout. Next we have the distribution. Before this they have three species of the Trypanosoma, that is gambiense, rhodesiense and crucial. Distribution, it shows the endemic in their distribution. If we talk about first, that is the Trypanosoma gambiense, so it is found especially in the central and west African countries like Nigeria, Zambia, Congo, etc. Okay, so this Trypanosoma gambiense causes more than 90% of the African sleeping sickness, that is the Trypanosoma gambiense. And if we talk about rhodesiense, so it is endemic to the southeast African countries, and T. cruzi is found in the South and Central America. Okay, next we have morphology. So this is the structure of Trypanosoma gambiense. So Trypanosoma gambiense, it has the unique characteristics that it shows the polymorphic flagellate protozoans, and it occurs in the four forms we will see in this polymorphism. Okay, so trypanosome or trypomastigot is considered as the adult form. So this is the adult form. If we talk about its shape, we can see that it is a flat and wavy body, approximate 2.5 to 10 micrometer in width and tapering at both the ends. Okay, so it is the transparent and asymmetrical. If we talk about its body structure, so pellicle and the undulating membrane here is present in body structure. Pellicle is the outermost thin, elastic and firm membranous fold, that is the undulating membrane. If we talk about its function, so pellicles maintain the journal shape. It will maintain the journal shape of Trypanosoma, while the undulating membrane, it helps in the locomotion. Okay, so it helps in the mobility, where it helps in the mobility in the blood, lymph and the cerebrospinal fluid. We earlier discussed this is first initially found in blood, then lymph and in the cerebrospinal fluid. How they migrate? They migrate with the help of undulating membrane. Okay, next we have the flagellum. This is the free flagellum uniflagellate. Okay, so this single flagellate arises from the basal granules. It arises from the basal granules, and it is the eukaryotic flagellum having 9 plus 2 arrangement. We studied 9 plus 2 arrangement is found in the eukaryotic flagellum. Okay, so similarly this type of arrangement is also found in the euglena. Okay, so if we talk about functions, so flagellum also helps in the locomotion. Okay, so next we have the cytoplasm. Okay, so there are the in the cytoplasm lies within the pellicles. Okay, but do not differentiate it into the ecto and endoplasm. So it is with the following structure, that is the kinetoplast and the cell organelles and nucleus. So kinetoblast, it is also called the paravasal body. Okay, so it is small, we can see it is a very small and spherical or discoidal body present near the posterior end of the body. So it will be small, spherical or discoidal body and present near the posterior end. This is the posterior, this is the anterior. So this kinetoplast and basal granules are interconnected. This kinetoplast and basal granules will basal granules will interconnected by the two fine fibers called the axoneme. Okay, So connected by the axoneme and the rhizoblast. So it is darkly stained. Kinetoplast will be darkly stained by due to the presence of extracellular DNA. So it has the DNA, so it can self duplicate. Organelles means it can self replicate, and this kinetoplast is modified part of mitochondria. Next we have cell organelles. So cytoplasm is with elongated single mitochondrion. There have the different types of cell organelles like mitochondria, Golgi body, and endoplasmic reticulums like RER and SER, and bulletin granules are present in this. If we talk about nucleus, so nucleus is single spherical well double vesicular and centrally placed nucleus. We can see that it is a spherical in shape and centrally located. Okay, it is located in the center and single nucleus. So it is also called the trophonucleus. It has double, and if we talk about nuclear membrane, so it has a double and porous nuclear membrane and a centrally placed, that is the endosome or nucleolus. Okay, this is the nucleolus, or we can say endosome. So this is the Trypanosoma gambiense. Now if we talk about electron microscope structure of Trypanosoma gambiense, so this is the structure. So we should draw the diagram. In zoology, diagrams are most essential part. Next we have polymorphism. So Trypanosoma shows the polymorphism. Polymorphic means having different types of polymorphological forms under the conditions. We can see this. Okay, that is the eumastigot. So according to the scientist Hoyer, it recognized the 6 morphological forms. Four forms is clearly distinct from one another on the basis of position of kinetoplast, basal granules, and nature of flagellum. So we can see the diagrams. So this is the leishmanial. In this fibril and blepharoblast and kinetoplast is located in this, but in this kinetoplast is located this in the leptomonad, and in the cristidial kinetoplast located in this. Okay, so location of kinetoplast, it shows the 4 types of polymorphic structures. So this is the Trypanosoma. Okay, so this is the leishmanial. In the leishmanial, body is oval or rounded, we can see, and position of basal granules and kinetoplast dot like, we can see it lie in the front of centric nucleus, and here is the flagellum produced fiber like and embedded in the cytoplasm. So it is called the amastigote. If we talk about the undulating membrane, so here is absent undulating membrane in case of the leptomonad. So it is the elongated as comparison to this. Okay, so if we talk about kinetoplast, so lies anterior end. Here is the centric and here is the posterior end of the body, and here is the plasma reduced fiber like and embedded in the cytoplasm. Okay, so here the free flagellum is present. Here is a long as comparison to this, we can see, and this flagellum is very large as comparison to this. Undulating membrane is present. Okay, so in this we can say inconspicuous present. Okay, so this is a very less, there is a very large type of undulating membrane, this absent, and the leishmanial undulating membrane also absent. Okay, next we have physiology. In physiology, we will see the nutrition, locomotion, respiration, and reproduction. Nutrition is the osmotropic in nutrition and absorb glucose of blood and tissue fluids to the general body surface. So it shows the osmotropic type of nutrition that will absorb the glucose from the blood and tissue fluids by the general body surface. So locomotion, it shows the swim. It shows the swimming nature in the body fluids by wavy movements. I earlier discussed undulating membrane and flagellum helps in the locomotion. Okay, so respiration, it shows the anaerobic respiration. Anaerobic means energy will be produced by the glycolytic pathway of the glucose, but it can also become the aerobe, but mostly it is the anaerobe type of respiration. Now we talk about the reproduction. So Trypanosoma does not undergo sexual reproduction. It reproduce only by the binary fission, that is the asexual fusion. So in this, kinetoplast and basal granules will duplicate. Okay, so all flagellum remain attached to the one basal granules. Okay, while the other basal granules will give rise to new flagellum. So nucleus will divide into two nuclei by the promitosis stage, and cytokinesis is along longitudinal axis and extend anterior posteriorly. Next we have the life cycle. We will see life cycle. It is a digenetic, that means it have two hosts. Primary host is the man and secondary host CC fly. Okay, so first is the human inoculation. We can see here, this is the life cycle. Okay, structure is must important. So first of all, human infection will take place. Human infection will indirect or inoculative, in which infective small-sized metacystic trypanosome forms with no free flagellum and are injected in the blood of man. So injected free flagellum will injected into the blood of man along with the saliva of blood-sucking insect. Okay. After that, multiplication of metacystic horns feed upon the glucose. We already discussed it. It shows the nutrition as a osmotropic, that means absorb the glucose of the blood and other tissue fluids through the general body surface. So here it will shows the locomotion by swimming type of locomotion, and aspires the anaerobically and multiply by the binary fission. After this, they will show the metamorphosis. Okay, metamorphosis means it forms the new form. Okay, like new form than larva. So firstly it will show the this type. You can see here it will develop here like this. Okay, next is the life cycle in CC fly. So transmission to the CC fly, this is short and stumpy forms are sucked by the vector host along with the sucked blood with the help of pursing and sucking type of mouth parts. So there is a short and stumpy, you can see here, this is okay. So this type will sucked by the vector host. We can see here the vector host. Okay, another type like CC fly. Okay, so CC fly sucked these infected forms with the help of pursing and sucking type mouth parts. Then here will development of midgut in the midgut of fly. You can see here. So these stumpy forms a parasitic double for about 14 days with the uh, peritrophic membrane of the midgut, and again transform into the long slender form. You can see this, and this also multiply rapidly by the longitudinal binary fission. During this development period, energy is derived by the aerobic respiration. This study here, it shows the aerobic respiration. Okay, so after that, development in the salivary glands takes place in the fly. So finally in the last step, so these long slender form reach the salivary glands through the esophagus and mouthparts of fly. Here these transform into the critidial forms. Okay, after that the dividing critidial and last metacyclic forms will generate. Okay, so the which is metacyclic form is the infective stage. So this whole cycle will takes place in the fly usually between the month, or we can say 20 to 30 days. Okay, so this is the basic structure of CC fly. Next we have the disease and symptoms. So trypanosoma is cause of fetal disease, that means it can cause the death. That disease is the trypanosomiasis, commonly called the sleeping sickness disease. So there are different species of trypanosoma as already discussed, that is the gambiense, rhodiensis and cruzi. So it causes the different variations in disease. Okay, different virulence. So like distribution, we already discussed its position, nucleus, and cause the disease and vectors of this. Okay, so mainly it causes the Gambian fever. These are the symptoms, and headache when inside the blood, and it causes the swelling of lymph nodes of neck. Okay, there is a swelling of takes place in the lymph nodes. Okay, so armate when in the lymphatic system. So these all are the like parasite damage the brain. Okay, the damage of brain can take place and it causes the sleeping sickness. So in this, fever, anemia, weakness, loss of weight, sleep. So these are the some symptoms of the trypanosomiasis. Next we have last, that is the prophylaxis or prevention. So killing the bacterflies by the various insecticides like DDT, BHT. So clear the brushes along the rivers which act as a resting sites of fly. Okay, so clear, it should be clear. And ingestion of 1 gram suramine, that is the antropol, after every 2 or 3 months. Or killing of reservoir host who is not so economical. Next we have some drugs which are used to treat the trypanosomiasis disorder, like where 205 ceramine sodium and germanin. So these all are the most effective drugs for use to treat the trypanosomiasis. So that's all about the today's video, and if you have any query you can simply comment in comment section. Thank you.